take a good look at this. This is gas gangrene. It's a medical emergency that spreads like wildfire through tissue. As a future doctor, you absolutely have to recognize this and know exactly what to do, because every second counts. Hey everyone, and welcome to our high-yield explainer on gas gangrene. We're going to cut right to the chase and focus on everything you need to crush your exams: the lab diagnosis, the key identification tests, and of course the critical treatment plan. So here's our game plan. We'll kick things off with the lab diagnosis basics. Then we'll zoom in on our main suspect, Clostridium perfringens, and learn two classic ID tests, the Nagler reaction and the reverse camp test. And we'll wrap up with the all-important treatment protocol. Let's get into it. All right, first up, the fundamentals of lab diagnosis. It all starts with getting the right sample from the patient and knowing what you're hunting for under that microscope. You know, getting the specimen is everything, and you can't mess this up. You can't just swab the surface, nope. You have to get deep into that wound, where the action is, and get samples of necrotic tissue or muscle fragments. And remember the rule of 3. You need 3 separate swabs. One for the direct gram stain, one for an aerobic culture to see what else might be growing, and the crucial third one for the anaerobic culture. Now, getting that sample to the lab safely is just as important. We use this special stuff called Robertson's cooked meat broth. So, why this broth? Well, Clostridium species are what we call obligate anaerobes. That's a fancy way of saying oxygen is poison to them. Even a little bit of air can kill them off, and you'll get a false negative. This medium creates a perfect, cozy, oxygen-free ride to the lab. Okay, samples in the lab. First stop, the gram stain. These are the usual suspects. Our number one target, C. perfringens, has this classic boxcar shape. They're big, gram-positive rods. Here's a key exam tip. In a tissue sample, you won't see spores. Now compare that to C. septicum, which looks more leaf-shaped and does have spores, or C. novii, which are large rods that also have spores. This table is gold for your exams. And here you have it. This is the textbook picture of Clostridium perfringens. Just look at them, thick, stubby, lined up like little purple boxcars. If you see this on a gram stain from a nasty wound, your alarm bells should be ringing loud and clear. All right, moving on. The smear gave us a big clue, but now we need to prove it by growing the bacteria. So let's look at the key culture characteristics that really nail the diagnosis of C. perfringens. Remember our Robertson's cooked meat broth? Well, it does more than just transport. C. perfringens is saccharolytic, which means it loves to ferment sugars. When it does that, it turns the meat in the broth pink. On the other hand, proteolytic species, like the one that causes tetanus, break down protein, which turns the meat black and makes it stink. So here's your super simple memory trick for exams. P is for perfringens, and P is for pink. Easy, right? Now, on a blood agar plate, C. perfringens does something really distinctive called target hemolysis. It's like a signature move. It uses 2 different toxins to create 2 zones of damage. There's a small inner zone of complete destruction, that's complete hemolysis, caused by its theta toxin. And around that, you get a much wider, hazy zone of partial destruction or incomplete hemolysis, caused by its infamous alpha toxin. And here's what that target actually looks like on the plate. See? It's unmistakable. You've got that small, clear inner ring, total hemolysis, and then the much larger, fuzzy outer ring. That's the incomplete hemolysis. Seeing this target on your plate is a dead giveaway that you're dealing with C. perfringens. Okay, on to section 3. That alpha toxin is the main weapon C. perfringens uses to cause all this destruction. So it makes sense we'd have a specific test to detect it, and that test is called the Nagler reaction. So how does this test work? It's all about what the alpha toxin does. It's an enzyme, a lecithinase, which means it loves to chop up a fatty substance called lecithin. So we grow the bacteria on a special plate with egg yolk in it, because egg yolk is packed with lecithin. When the toxin breaks down the lecithin, it creates this milky, opaque halo around the bacteria. The procedure is a classic control experiment, super simple. You take your egg yolk agar plate and draw a line down the middle. On one half, you spread an antiserum, basically an antidote with antibodies that neutralize the alpha toxin. The other half you leave alone. Then you just streak your bacteria across both sides, pop it in the incubator, and wait. And here is the result. It's beautiful. On the left side, the control side, you see that milky, opaque zone. That's the alpha toxin doing its thing. But look at the right side. Where we put the antitoxin, the effect is totally blocked. No milky zone. This is a positive test, confirming the presence of alpha toxin. But, and this is a big but for your exams, the Nagler test isn't perfect. It's not 100% specific. There are a few other Clostridium species out there that also make a similar toxin. So while a positive test confirms the presence of the toxin, it doesn't, by itself, definitively prove it's C. perfringens. Just keep that in mind.
So let's move on to another classic test that helps us get even more specific, the reverse camp test.
The principle here is all about synergy, two things working together to create an even bigger effect. You've got the alpha toxin from our suspect, Clostridium perfringens, and a protein called camp factor, which is made by group B strep. When these two get close to each other on a blood agar plate, they team up to cause way more hemolysis than either could alone. And the setup for this is super easy. You take a blood agar plate and make one straight line down the middle with your unknown Clostridium. Then you streak a line of group B strep across it, at a right angle, but make sure they don't touch. Then you incubate it and look for the magic, where the two almost meet. And this is what you're looking for, a perfect, beautiful arrowhead-shaped zone of super strong hemolysis. And notice how it points right at the C. perfringens streak. This synergistic clearing is a positive reverse camp test, and it's another huge piece of evidence pointing to C. perfringens. All right, last section, and it's the most important one. We've done our detective work in the lab. Now, what's the actionable plan to save our patient's life? How do we treat this? To make this really easy to remember for your exams, just think of the word DASH. D is for debridement. This is, without a doubt, the single most important step. A surgeon has to go in and cut out all of the dead tissue. A is for antibiotics, usually a powerful combo of penicillin and clindamycin. S is for serum, giving the patient antitoxin to neutralize the poison. And H is for hyperbaric oxygen, putting the patient in a high-pressure oxygen chamber to kill those oxygen-hating bacteria. So as you can see, this is a multi-pronged attack. You have to hit it with everything you've got, all at once. It's about aggressive surgery, high-dose antibiotics, antitoxin, and hyperbaric oxygen. There's no time to waste. You have to be aggressive, and you have to be fast. And this brings us to the single most important slide, the one thing I want you to burn into your memory forever. You do not wait for the lab to confirm your suspicion. Gas gangrene is a clinical diagnosis. The second you suspect it, based on how the patient looks and how the wound appears, you dash. You start treatment. The lab tests are for confirmation, yes, but the patient's life and limb depend on you acting immediately.